So this week we're going to look at the development of the heart and the vasculature. And we'll start with a quick review of the anatomy of the heart. Interestingly, the heart is actually the first organ that functions in humans, and that makes sense because the circulation of blood, the pumping of blood around the body, is critical not only for normal function of the developing embryo, but also drives some of the developmental processes that come after. So I know you watched the video. This is just quick review so that we're all on the same page for the lecture. The location of the heart. So of course your heart is located in your chest, but more specifically it's within the mediastinum. Now the mediastinum is not actually a cavity in the classic sense. It's not a space enclosed by membrane. It was really just the term for the place in between the two lungs. So your heart is situated between your lungs in a place called the mediastinum. The heart is also surrounded by a membrane, which is called the pericardium or the pericardial sac, two terms for the same thing. But the presence of that sac, that membrane, defines the pericardial cavity. So think of it a bit like Russian nesting dolls. The heart is within a membrane called the pericardial sac. Within the pericardial sac, that space within the sac, which is mostly heart, is the pericardial cavity, and then the pericardial cavity is within the mediastinum. Now, on the lower end, the caudal end, the bottom of the heart is bordered by the diaphragm, which we covered last week. And then, of course, there are ribs and muscles that surround the heart that help to cushion and protect it. Okay, so I made this diagram here mostly to serve as a reference because a lot of the diagrams that you see are somewhat more anatomically correct, but not as good at showing actual function. So it is important, and we will go over this several times this week, but it is important to understand how these diagrams are coded. The red vessels, okay, and the regions of the heart that are colored in red are showing regions of oxygenated blood. So that's going to be blood that's recently arrived from the lungs. So in normal circulation, blood flows through the lungs, picks up oxygen from the air that you've breathed, and then heads back into the heart, okay? And then the red blood is going to then head out to the body. That's its ultimate destination. There are two different paths, the upper body and the lower body, but that's where it's going. Whereas anything shown in blue, be they vessels or chambers of the heart, contain deoxygenated blood, the blood that's actually heading back into the lungs to pick up oxygen so it can return to circulation. Now, we'll go over this later. Arteries and veins, often arteries are oxygenated and veins are deoxygenated. So usually arteries are red and veins are blue, but there are exceptions to that. And we will talk about that when we talk about the vasculature. But always, 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 when looking at diagrams of the heart, red is blood that contains high quantities of oxygen, blue is deoxygenated blood. The other thing to know here, well, a couple other things. So the blood pressure is highest as the blood is exiting the heart, and that makes sense because the pumping action of the heart is forcing the blood out into the system, and it takes a fair amount of pressure to keep it going all the way to the organs and tissues that it needs to reach. And then, of course, the blood pressure in the venous system, which is primarily deoxygenated, is quite a bit lower. But all of the valves you see in the heart are one-way valves. They don't allow blood to flow backwards. So one of the ways that you keep blood heading back to the heart in the lower pressure venous side of the system is through these one-way valves. So even if the pressure drops a little bit and the blood isn't still flowing as quickly forward, it also can't flow backward. Feel free to use this diagram as a reference. I find it helpful. If not, then ignore it. Okay, the heart wall, the histology of the heart. So as with all of the other organ systems that we've discussed, the heart wall has layers. First, the endocardium, which is the innermost layer, and it's the sort of the lining of the interior of the heart chambers. Next up is the myocardium. That's the muscle. The heart is primarily a muscle. It's a giant pump, and so it has a lot of muscle, very thick muscle, thicker in some places than in others. But the myocardium, the muscle, is really what drives the primary functioning of the heart. The next layer is the pericardium or the pericardial sac. We talked a little bit about that a couple slides back. It has a couple different layers itself. So there's the serous pericardium. That contains the epicardium, which is sort of part of the heart wall, then this pericardial cavity, then the parietal layer. The pericardial cavity it's a fluid-filled space, and it's actually critical to heart function because it reduces the friction between contractions or during contractions. If you were experiencing high levels of friction, your heart would not pump as efficiently. So having a fluid-filled space there is critical for normal heart function. And then the parietal layer just helps sort of fuse to the fibrous portion of the pericardium, which is connective tissue, and it helps with positioning the heart within the chest. Okay, so on to the early development of the heart, and we will go through this over the course of a couple lectures, but we will start here. So heart fields, okay? The cardiac progenitor cells, so the cells that are going to go on to become ultimately the heart, they differentiate from the mesoderm and they migrate through the primitive streak. And if that sounds familiar, it should, because it gets us back to this, okay, which we looked at, I think, back in week 3. So this is back when lots of mesodermal cells and mesodermal cell derivatives are migrating through the primitive streak, and the cardiac progenitors are some of those. So they go through the streak and then to the left and the right, forming the left and right heart fields. And now those fields are actually going to sort of grow or migrate cranially until they meet at the top, forming a sort of upside down U shape or a crescent shape called the cardiac crescent. And that's your first heart field. Okay? The second heart field, and there are 2, exists on either side of the notochord, okay, and then also sort of migrates cranially, creating in parallel, pardon me, with the first heart field, okay? So you have these 2 heart fields, and you'll get actually major structures of the heart from both of them. Okay, so the heart tube. We're going to head back to body folding here in an effort to sort of show you where all of this comes from. But a couple quick reminders. So at this stage, okay, looking at this diagram, so the cranial, caudal, and lateral folds, and it's been a while since we talked about those, but basically it's folding sort of at the future head region towards the connecting stalk, folding at the future tail or rear towards the connecting stalk, and then the lateral folds the sides also towards the connecting stalk, okay? And again, the lateral folds are the ones that sort of create the gut tube, which we talked about earlier, okay? So the cranial end of the first heart field is moving ventrally and caudally, okay, so towards the stomach and the tail. Then the 2 sides of the first heart field are brought together by the action of the lateral body folds. So just like they're bringing the gut tube together, they also bring these heart fields together. Now, within the first heart field, on either side, so remember it's a crescent shape, so there's parallel sections of first heart field running down the length of the embryo. So within each side of the first heart field, you get a space or a tube that forms within each side, and those are the endocardial tubes. And those tubes ultimately are going to be brought together and fused to form the primary heart tube. And so even though your heart looks a bit like a house with 2 floors and 4 rooms, in reality it starts out as a tube. And I think that's kind of the take-home message of human embryology. It does seem that everything starts out as a tube. Okay, now, so heart tubes running parallel either side of the body. Now, cranial body folding is going to move the primary heart tube into the thoracic region, into the chest. Okay, so we're going to get it localized to the right spot. And then from there, some splenic mesoderm is going to surround the endocardial tubes, okay, and they will become the myocardium. So remember, that's the major muscle layer of the heart. The myocardium is then going to produce cardiac jelly, which is going to separate the myocardium from the endocardium, okay? And that's sort of step 1 for forming the heart tube. Now, keeping in mind that during all of this process, cell proliferation is continuing, and it's producing the more caudal regions of the heart, all right, so the lower regions of the heart. But we're dealing right now primarily with this sort of cranial region, primary heart tube. Okay, so now once we have the tube in place, the heart is going to sort of alternately, in some regions, constrict and in other regions expand, and that's going to define the chambers of the heart. And if it helps at all, think of it a little bit like those long tubular balloons you use to make balloon animals. If you twist them a little bit, and of course the heart isn't twisting so much, but if you cause constriction in certain areas, you get expansion in others. And that's what this is all about. All right, so we can now, from the heart tube, start to see the early stages of the basic structure of the heart. So we have the primitive atrium, which again, we ultimately are going to have two atria, but we start with just one atria, one ventricle that will then divide into two. And I should say there are a lot of things going on here in the heart anatomy. It's very complicated. We do not have time in an 8-week course to cover all of the glorious detail of heart development. So I'm going to try and focus in on the things that are really key, the things that are helpful, hypothetically for somebody practicing as a genetic counselor. But there's more to it, and there are some things that we're going to gloss over or skip entirely. Okay, so atria, of course, are important. So we have the primitive atrium, and then we have what's called the atrioventricular canal. Really, this is just the space between the primitive atrium and primitive ventricle. Okay, but it's defined on the surface of the heart by what's called the atrioventricular or AV sulcus. Anytime you see AV in heart, it's atrioventricular. So the AV sulcus is what's visible from the surface of the heart, underneath of which would be the AV canal. Okay. Important to keep in mind, the AV canal and its normal development is critical for function of the heart, and it's a developmental process that tends to go awry in children with Down syndrome. So classic for kids with Down syndrome are AV canal defects, heart defects derived from abnormal development of the AV canal. And we'll talk a little bit more about that in later lectures. Okay, but we also have the primitive left and right ventricles. There's not much defining them on the surface, but inside there is a muscular septum that's starting to develop between the two of them. And then we have the outflow tract. Okay, now the outflow tract is sort of defined by two sections, the proximal and the distal section. It will become the vessels that flow out of the heart. But the proximal section is going to end up joining with the ventricles and becoming part of the body of the heart. But the distal outflow tract, so the portion of it that's furthest from the primary heart tube, that is going to divide into the ascending aorta and the pulmonary trunk, two huge vessels. And in fact, another name for the distal outflow tract is the truncus arteriosus. And if you remember back to week 2, when we were talking about neurulation, we talked a little bit about a heart defect called truncus arteriosus that comes from not being able to split your ascending aorta and your pulmonary trunk into two separate vessels. Okay? That defect would occur at this early stage of heart development, and actually we'll pinpoint exactly where it happens in a later lecture. Okay. In fact, we might cover it right now. So, in here, yes, pardon me, we're covering it right now. I'm sorry, guys. I lost track of where I was at. But this is a reminder back to that week. We looked at this picture before, and what you're seeing here is that, so AO would be your aorta, and PA would be your pulmonary artery or pulmonary trunk. They should be two separate vessels. We'll talk later about how they get into that sort of twisted helical position. But if they don't divide into two, then you have a single outflow tract that feeds both ventricles. So we're mixing oxygenated and deoxygenated blood, which is never a good thing except in a fetus, and we'll talk about that later. But it's not supposed to happen after birth. And so actually what we're getting is not enough oxygen flowing into the body because it's mixing with deoxygenated blood and flowing out through the same trunk. Okay. So lastly for this week, or pardon me for this lecture, the primary heart tube, again, as we talked about, it's derived both from the first heart field and the second heart field. And it's worth paying attention to which things are derived from which field. The first heart field derivatives include both the atria, the atrioventricular canal, and the left ventricle, but not the right one. The second heart field produces the right ventricle and the outflow tracts. And so actually, I've got them highlighted on the upper diagram here, but if you look in the bottom one, and this of course all comes from your book, they've got it color-coded the same way that I have here, and you can actually see in the final arrangement of the heart that the second heart field contributes to a fairly significant portion of the final heart. Oh, and one last thing before we finish, sorry guys, cardiac looping. All right, so we're going to spend a lot of time talking about how this tube sort of curves in on itself and things twist and turn, and the heart is just a whole tango in and of itself. But what we do see is that this sort of process of growth and elongation, so certain sections are growing, other sections are sort of stretching, some are doing both at the same time. And what that's doing is forcing that differential growth, forcing portions of the heart together, so forcing the tube to curve and then forcing portions together, and that's ultimately what's orienting these heart chambers in their final adult positions. Of course, we'll talk a lot about how we get septation, how we get separate chambers, that's a separate sort of thing. But this process of the tube curving, looping, coming back up on itself is what puts those regions of the tube that will become the future chambers and outflow tracts into their final